last lecture we are discussing about the diversity of B lymphocyte and T lymphocyte and what is the maximum number we can reach in different diversity of two different lymphocytes. Now, today or in this lecture actually we are going to talk about structural variation in the immunoglobulin constant region. Okay. So, and later on we are going to talk about how this isotype is switching, because different constant region of immunoglobulin are known as isotype, right. So, any particular antigen, if it is recognized by the hypervariable region or the variable region of antibody, its constant region may be IgG type, IgM type, IgA type, but the specificity towards the antigen is specific. So, against any particular antigen, what can happen? One antibody binding site can hold it, okay. But its constant region, which is responsible for the effector function, can be IgM or IgG, IgA, IgE, right. So, different type of constant region is possible. So, today we are going to talk how this structural variation in the constant region is generated. Okay. So, this particular slide you already have seen the upper part, right. When we are discussing different type of antibody or antibody structure, we showed you that IgM, IgG, IgD, IgE, IgA different. So, IgM, IgG, IgD, IgG, IgE, IgA, varieties of these 5 different isotypes of antibody we have discussed. Its structural difference little bit also we will discuss, but today what we are going to tell about that how this constant region is coming to a particular type of variable region. We have to remember that constant region is of antibody is only contributed by the heavy chain, right. So, if we see how this heavy chain is rearranged, then we can tell what can be the isotype of the antibody. So, if you see below, just below this antigen antibody structure, you see there is a series of same color code boxes. What these boxes? This is basically the constant region gene segment. Okay. Now, this is the upper panel is mouse and lower panel is this is the human one. So, now if you see the mouse and human, both cases JH is there. So, what is there before that, or the 5 prime end of this gene, this is the DNA strand. So, 5 prime of this JH we had D and before that we had V. So, after the J region we have different constant region. If you see here, the constant region is designated as C and suffixed by one Greek letter. What are those Greek letter? If you see this mu, delta, gamma 3, gamma 1, gamma 2b, gamma 2a, epsilon and alpha. You can easily correlate. I mean, if you see the name of the antibody gene like isotype, IgM is coded by C mu, IgD coded by C delta, IgG is coded by gamma, and there are different subtypes. If you see gamma 1, gamma 2a, gamma 2b and gamma 3. So, there are 4 different subtypes of IgG, and then we have IgE, and then IgA is coded by C alpha. So, what is happening after this VDJ recombination? VDJ recombination is over, and after that it is giving the only variable region. So, after variable region, constant region is coming. Which constant region is going to come? It depends, I mean depending on that what isotype will be the final product is determined. Okay. So, VDJ recombination in heavy chain, VDJ recombination in heavy chain is common. After that, if mu comes, then it will be IgM, if delta then IgD, if gamma then IgG, if epsilon then IgE, if alpha then IgA. If you see the human genome and the arrangement of this constant domain, they are almost similar, okay, but there is two alpha subtypes, alpha 1 and alpha 2. Okay. There one you can see here that psi C epsilon. I do not know how many of you are aware of this psi designated gene. Psi actually, psi before the genome, I mean before any gene segment of gene indicates it is a pseudogene. Okay. There are many pseudogenes present in human genome. Pseudogene actually, the gene, I mean this is not related to here, but just for your information, pseudogene many of you may know already, pseudogenes are the gene which does not express, but prediction, I mean if you see their sequence, it is a complete gene. The two possibility, immediate two possibilities you can find, like why one particular gene is present in chromosome, but is not expressing, because gene expression first thing you need, it should be transcribed. So, for transcription you need a promoter, right, and if promoter is not there, it is not going to transcribe. That is also called pseudogene, because gene is there, but it is not transcribing, that is why no protein. Another possibility is that even the promoter is there, but there is maybe a stop codon inside, so that we will not get the complete protein product, it will be truncated. So, final product will be incomplete protein or no protein. Okay. So, that is also maybe the cause of pseudogene. So, here this psi C epsilon is pseudo, otherwise all are functional gene. Okay. Clear? So, this table actually is very, I mean you do not have to remember, do not be scared, you do not have to remember all these figures, but this table if you go slowly, I am not going to spend much time on it. This is actually saying that which is the heavy chain, like IgG1, Ig2, G4, IgM, IgA1, A2, IgD and Ig. You can understand this is human one, because IgA1 and A2 basically is all subtypes of human, not present in mouse, and this is their molecular weight. I am going to tell I mean molecular weight, how you can estimate that, or if you want to do verify that what I am telling or the written in the book is right or wrong, then you can do some very simple experiment to check that. So, these are the molecular weight. So, normally what we say is the average molecular weight of IgG is 150, just to be it is not exactly. So, IgG has different, you see IgG 1 is 146, 2 is 146, IgG3 is 165. So, average is close to 150. So, IgG molecular weight is 150 kilodalton, this is in kilodalton, and if you see IgM, which is huge, but if you go back, I mean if you see the structure, IgM only had one extra constant domain. So, one extra constant domain is contributing only 110, approximately 110 amino acids. So, these 110 amino acids will not contribute that much. So, why this is suddenly so high? It is almost 5 times, that we will see later. Okay. And Ig is little more, because it also has one extra constant domain. So, their serum level, if you see that maximum serum level, or at any time an adult in if you check this isolate the blood and purify the serum, and in serum if you measure how much IgG is there, then it is the maximum amount. Okay. And IgM is also good, IgA is also not I mean in between, but there are others are not as much. Okay. Their half life is also given. So, IgG has the maximum half life. So, from this what we can predict is, we can predict that most of the adaptive immunity what we are seeing, or what effect we are enjoying actually the immune system, that how they are protecting, major player in antibody mediated immunity is played by IgG. That is why they are more in amount, they are more in their half life is also more, that means they stay in blood for longer time. Okay. So, IgG is one of the very important antibody isotype, which plays a major role in adaptive immunity mediated by antibody. And bottom part, if you see, bottom part has the effector function. Some of the effector function we already discussed, like opsonization, neutralization, complement activation. So, you see this plus and minus, which indicate plus means it is helping in, say for example, classical pathway complement activation. Best performer of this activation is IgM, you see 4 plus, and the number of plus indicating how good it is, or and negative means it does not take part in the complement activation. So, you can see only IgM and 3 of the IgG are mostly taken care of this complement activation. IgM is the best player and IgG2 is the least efficient player. Alternative pathway complement activation, that this is you do not have to remember right now, but you learn when the complement will teach complement system of immunity, then you will understand what is this alternative pathway. Only IgA1 is involved. Same way, if you go through this, then you can understand which antibody is doing what. And here is one thing which we did not discuss, placental transport, which is very important, because when baby is growing inside mother's womb, then mother's blood is actually protecting the baby inside the womb, because that time immune system is not ready, their antibody production, their exposure. So, any kind of protection of a baby inside the mother womb is protected or taken care by the antibody present in mother's blood. So, if antibody cannot cross the placental barrier, then it cannot help. So, not all antibodies are crossing the placenta. So, there are few like IgG1, and you can see from here, right. So, and gradually you can see what is happening. Here at the bottom, one thing is written, that reactivity with staphylococcal protein A, reactivity with staphylococcal protein A. Okay. I will come to that, not exactly in relation to the infection, but we will discuss, or I will discuss what this protein A can do and how we use this protein A. Protein A actually the surface protein of Staphylococcus aureus. Okay. It has a very good property which is not good for health, but the technologist or the scientists developed some technique to purify the antibody, that we will discuss sometime if time permits. So, this reactivity with the staphylococcal protein A is, you see, mostly the IgG are interacting with staphylococcal protein A. So, what it is actually doing, this protein A is the surface protein of Staphylococcus, they have a specific property, it can bind to the constant region of IgG. Okay. Constant region of IgG. So, by that they can eliminate the IgG to do its proper function. Okay. This is a defense mechanism from bacterial side. So, now we can see from this table, most of the antibody present in our serum is IgG, and staphylococcal antigen can clear them by binding them. Okay. So, IgG actually cannot do much to protect from Staphylococcus. So, that property, that means protein A binds to IgG, is also exploited to purify the antibody, that we will see if time permits. Okay. So, now different classes of immunoglobulins are distinguished by the structure of their heavy and constant region, and this constant region confers the functional specialization of the antibody. That I am just, this is the summary of the just the last slide. So, different classes of immunoglobulins are distinguished by the structure of the heavy chain, that we already have seen in last few slides, and the constant region confer the functional specialization, like which one is going to do the complement activation, which one will do the opsonization. The last table, the bottom part is actually saying by this statement. Now, IgM and IgD. Okay. IgM and IgD are derived from the same pre-mRNA transcript and are both expressed on the surface of mature B cell. I am repeating again, though it is written, IgM and IgD are derived from the same pre-mRNA. How it is possible? So, before going to go there, what I will do is, what I am going to do is, in B cell, if this is the B cell, before the antibody is secreted, what which form it is present in the surface? It present as a receptor. Okay? It is present as a receptor, and upon activation the same receptor are synthesized in different form and secreted. Okay? So, what is there in the which form of the isotype is present as a receptor? Actually, they are mostly IgM. Okay. And they also have IgD. Normally IgD concentration in blood is very low and they do not secrete. Okay. So, IgD actually take part in the initial signaling during the activation of B cell. This is B cell or B lymphocyte. So, during the activation of B cell, that means when B cell is there, receptor is there. So, this is the receptor in B cell, and if antigen comes, it binds, and which gives the signal to the B cell that okay, something you have to take care. And along with that signal, signal from T helper cell is also giving the signal to the same B cell. So, when two signals comes together, that we discussed in the during basic concept of immunology, in the first few lecture we discussed. So, in B cell actually two type of receptors are there, two isotypes of receptors, same specificity or antigen specificity of these two IgM and IgD both are same, but only their constant domain are different, and both of them attached to the B cell as a receptor. IgM and IgD both interact with the antigen and gives the signal to B cell to proliferate and to transform to plasma cell for production of antibody along with the signal from T helper cells. So, how this IgM and IgD always come together? It is saying that IgM and IgD are derived from the same free RNA. Now, we will see what is happening, how it is located. So, if you see, this is the structure actually what is happening, do not be scared, it is very simple. So, if you see the structure or the organization in the genome, you see C mu and C delta is side by side. C mu and C delta both in case of mouse as well as in case of human. I do not remember whether I told previously in class or not, but most of the immune system what we are studying, what we understood so far, much of them or most of them are studied in mouse, because mouse immune system and human immune system are very similar, not exactly identical. There are there are many places where they are different, but most of the cases they are very similar, even in the gene organization also. Okay, but we cannot do all this thing in human. So, mouse is very good model to study human immune system. You see here, this case C mu and C delta organization in mouse as well as C mu and C delta, that means IgM and IgD constant domain organization in this human are exactly identical. So, what is happening? How the C mu and C delta always comes together, that we will see. Okay. So, here if you see actually how this, if you just enlarge that C mu and C delta part, how it will look. C mu and C delta part, VDJ is already recombined. Then immediately after that, this is the DNA. I told you once and I am reminding you again that when there are two lines between these boxes, that means DNA, and when there is one line, that is RNA. Okay, because of just in cartoon, and also signifies the double strand and single strand. So, VDJ is next to VDJ is the C mu. In previous slide C mu was only one box, but here actually this CH1, CH2, CH3 and CH4. There are 4 domain in C mu, and this region in between is a hinge region. Okay. But in other case, but here there is no hinge, because in IgM there is no hinge. So, this is C mu, and there is a 1 PA1. PA1 is what? In eukaryotic system, you know, in eukaryotic system there is a, in every mRNA there is a polyadenylation site. That means poly A has been added in transcript after in the mRNA, and the eukaryotic mRNA contains poly A tail at the 3' end. So, it needs a signal, there is a site, specific site in the genome also. So, after this C mu there is one poly A signal, then immediately after that this is the C delta constant domain. In C delta this is CH1, this is CH2, this is CH3, this is CH3, CH1, CH2 and CH3, and these boxes actually talking about the hinge region. Okay. So, in previous slide what we have seen that C mu and C delta are next to each other, but this one box is not really one box, this box is actually are several domain of C mu. So, now what happened during expression, you first you need to have a transcription. So, when it is transcribed, it transcribes the whole thing. So, VDJ, C mu and C delta transcribed together. Okay. So, this is that whole transcript, VDJ is common, then C mu and C delta. In this, after this transcription happens, this is a precursor RNA, you know precursor RNA undergoes splicing, and after splicing the mature mRNA come, and this mRNA is the responsible for what will be the protein sequence. So, this is one RNA sequence. Okay. This RNA sequence is having one VDJ plus C mu segment and C delta segment. In one splicing, what is happening? VDJ is there and then only C mu is spliced. As there is already a poly A signal, so what is the mRNA? mRNA have only C mu part, which is all this internal intron type, intron are spliced out and they come together. So, product of this mRNA is actually IgM. Okay. And this is actually a very good example of alternate splicing. Okay. If you do not know what is alternate splicing, please go and check that what is alternate splicing, but in there are many genes are going to undergoing the alternate splicing and giving two different product of two different protein. So, in very brief, alternate splicing is, suppose there are two exons, okay, let me change the color, there are say exon 1, exon 2, exon 3. So, if this is the precursor RNA 1, 2, 3. So, in one splicing, suppose exon 1 and exon 2 come together, okay, 1 and 2. So, these two splice together and they are one poly A. In other one, if it is possible, I am going this way, what happened? Exon 2 and exon 1 join. So, as a result, you will find exon 1 and exon 3. Okay. So, what is going to happen? So, there is only one mRNA. So, one protein is having 1 and 2, another protein is having 1 and 3. So, though there are one mRNA, but you can have two different protein of two different sequence. From this is alternate splicing. So, here we are going to see the same, we are going to see the same example, I mean same alternate splicing here. So, one mRNA, one precursor RNA which have both C mu and C delta as well as VDJ. So, if I consider the whole thing, I mean there are so many introns and exons, but I am, so to make my life easy, say VDJ is 1, C mu is 2 and C delta is 3. Here one is what is happening, 1 and 2 is combining and giving IgM. Okay. Another case, what is happening, 1 and 3 is coming, 3 means C delta constant domain. So, 1 and 3 is making IgD. So, one mRNA. So, they are so close in the genome, one promoter and one product. So, as a result, if we go back to this slide. So, as a result, in B cell as a receptor we are seeing IgM and IgD is together. Okay. As a result, IgD and IgM is coming. This is the alternate splicing product of single mRNA, which is giving expression of IgM as well as IgD. Okay. Now, question, I mean, I hope it is clear. Now, I will explain the very similar thing. Now, I told several times that same receptor molecule, this receptor molecule is synthesized as a secretory protein upon activation. So, same receptor molecule secreted as antibody molecule upon activation of the B cell. How this thing happen? Because receptor has a transmembrane domain and secreted protein antibody does not have that transmembrane domain, otherwise that cannot be secreted. Here is again similar alternate splicing is happening. Now, we have to see more detail of that gene. In the previous slide it was less detail. Now, if you see closer look of the IgM, okay, what we will see? We will see VDJ is there, we will see CMU1, CMU2, that is CH1, CH2, CMU1, CMU2, CMU3 and CMU4, all are as usual before, and then what was not there in the previous slide, there is one SC, one M1 and one M2. Okay. SC, M1 and M2. SC is for secretion, which is a tail, but not transmembrane, and M1 and M2 is basically contributing the transmembrane domain of the receptor antibody molecule or B cell receptor molecule. So, here again alternate splicing is happening, one product. So, this is DNA and one single mRNA is happening. Here you see there is one PAS and PAM. This PAS and PAM both are polyadenylation signal. Okay, so, transcribe, after transcription everything is coming, okay, everything is here. But so, in one product what is happening? VDJ is coming, all this C1, CMU1, CMU2, CMU3, CMU4 all are coming, okay, but this SC portion is not coming. So, it is not spliced. Instead of, I mean, in after that this M1 and M2, which is contributing the transmembrane domain of the B cell receptor, are joining. So, all these are different exons. Okay. So, in first case this SC is not coming, instead of that 2 YOLO, which is the M1 and M2, that the transmembrane domain is coming, and you can see this YOLO is indicated as a transmembrane domain part, which is a carboxy terminal. So, as a result what is happening? This is the final mRNA. So, this product if converted to protein, which will have a transmembrane domain and which will remain attached to the membrane, clear. But the same transcript is happening upon activation in plasma cell, but splicing is happening differently. What splicing is happening here? Here is what is happening. Instead of M1 and M2, this SC portion, which is secreted IgM portion, which is not as big as this whole transmembrane domain, but they have a little tail, okay, and this PAS, which is the polyadenylation signal, is coming. So, here what is happening? Same VDJ, same IgM constant domain, but instead of M1 and M2, this SC portion is coming. Okay. So, what is the take home message from here? One mRNA, multiple exons, out of that VDJ and all constant domain region 1, 2, 3, 4 are always there. In one case secretory domain is there, in another case transmembrane domain is there. So, normally in B cell, when it is expressing not activated, not antigen is there or not converted to plasma cell, they are expressing the gene along with the transmembrane domain. So, they cannot leave the cell and remain attached to the membrane or plasma membrane. That is why it is the receptor and doing its job, and this carboxy terminus or the cytoplasmic tail part is responsible for signaling. Okay. So, upon activation, same gene, everything is same, but just a slight change in the splicing make the same molecules to secretory protein, that then it secretes to blood and doing its own job like neutralization, opsonization, complement activation and many other things. Clear? So, but if you see, the variable region is not changing. Okay, that what we say, that every B cell will produce a single type of antigen specificity, because once one VDJ recombined give a specific specificity against a particular antigen, that remain constant, it is not changing, and the same molecule or same VDJs also act as receptor, also secreting as antibody. So, there is no change. So, throughout the life cycle of the one B cell, it may change by hypermutation, but once it is changed, this VDJ will remain same. So, same molecule is going to be receptor as well as antibody molecule. Okay. So, this is the reason why this particular receptor molecule converted to antibody without changing its specificity. So, this is the mechanism, or this is how it is happening. Okay. So, I will go quick. Two things. One, why IgM was so high molecular weight? Because IgM always stay as pentamer. So, 5 molecule, if you see antibody is like 1, this is IgG, but IgM always remain as 5 together, the pentamer. That is why it is molecularity is so high. And this is a very common question, how in pentamer IgM, how these monomers are attached, peptide bond or what? No, you see all are disulfide bond. Even the J chain which is connecting, this J chain is also connected to monomers by disulfide bond. Similarly, IgA remain as dimer, same J chain as and disulfide bond. IgA remain as dimer and IgM remain as pentamer. Okay. So, I am repeating again, IgM as pentamer, IgA as dimer. So, if you remember what is the valency of antibody, normal IgG valency is 2. What is the valency of IgM? So, IgM valency is 10, because each one can bind 2. So, valency of IgM is 10, valency of IgA is 4, other like IgG, Ig all have valency 2. Okay. So, we will see you in the next class. Thank you very much.